What is today's date? 18. Am I audible, guys?
Yes, sir.
Yeah. So we finished carbohydrate completely, and now we are supposed to start the protein part, correct? Because we know protein are the most abundant biomolecule compared to the carbohydrate, lipid, nucleic acid. So our next topic is protein. Okay. So first point, protein, one of the vital structure, important structure that require in the highest amount. Why? Because if you read in the uh, biomolecule of biology, then it is written that 10 to 50% of our body is protein. When people eat chicken or meat, they are looking for more protein, correct? So that's why they like chicken have high dressing percentage. That's why protein is the most abundant biomolecule in the living system. And when we are saying biomolecule, it means we are comparing with the carbohydrate, lipid, and nucleic acid. Correct. First, what is the name? Sania, what is the source of the protein? Milk, cheese have protein, pulses, we eat peanuts, we eat fish, we eat meat. All these are the source of the protein. Correct.
Yes, sir.
And why it is important, like why we want to know about this? Because sometimes they can give you which of this food contain maximum amount of protein, and option will be like rice, ragi, wheat, and one option is peanut, like this. So they can give one statement from the NCERT. Hello. हाँ. के दिखा भाई, के दिखा भाई, फिर पाया. चलो ठीक है, फिर मैं करूँ. तेरे तो थोड़ा ये नहीं आ जाएगा. चलो कोई नहीं, मैं नरवाने तो ले आऊंगा, फिर नरवाने तो ओके. ठीक ठीक. हाँ भाई, इसमें जो ही शरीर खराब होने लगे तो क्या मतलब है इसने किसे बताना. नहीं नहीं, मैं रिसर्च करूंगा, गूगल पर रिव्यू देखूंगा, सारी सारी चीज देखूंगा, रिसर्च करके करूंगा. ठीक. अच्छा. Okay. So that's why, though you know about all these things, but still we need to mention because it is given in the NCERT. Move to the next one. When it comes to the protein word meaning is primary importance. Why it primary importance? Because we require in the maximum amount. The word protein meaning is primary importance. And why? Because it requires in the maximum amount. So, uh, Chandana, what is the monomer of the protein? Chandana,
The
one
amino
more.
acid.
Alpha amino acid. Sarah, why we call it as amino acid? Sarah, why we call it as amino acid?
So because it contains an amino group and a
And
carboxyl.
yes, because this contain one amino group, one carboxylic acid, one hydrogen, and one variable structure. This variable group will decide which kind of amino acid do we have. Approximately, we have around 20 amino acid, 20 types of amino acid. And when we are saying 20 types of amino acid, it means there we have 20 different R groups. It could be hydrogen, it could be CH3, or it could be anything else. Samar,
Yes, sir.
uh, do we have only alpha amino acid or other possibility are also there?
Sir, if there are longer carbon chains, it is possible.
Longer carbon chain, it is possible. No, we have only alpha amino acid. Okay,
Okay.
only alpha amino acid. No other amino acid is present in case of protein. Chinmay, am I audible to you?
Sir, but
Because I
other
think
amino acids can exist, sir. Like not in
yes,
protein.
but in case of protein, only
Okay.
alpha amino acid exists. Chinmay,
Okay.
you can write if you can't speak. Yes, okay. I think you can't speak, but it's okay. You, all of you have to write notes, and I'll check in the end of the class. So, Chinmay, what I did in last 2 minutes, कि protein are the most abundant biomolecule compared to carbohydrate, lipid, and nucleic acid. Present in all these food, you know this. Protein meaning is primary importance. Protein. So, primary importance. And protein are polymer. It means it is a big mass. Polymer of something small unit. And this small unit is alpha amino acid. How many type of alpha amino acid we have? 20 type. Do we have only alpha amino acid? Yes. Remember, only alpha amino acid. And you will be confused when you see this word only, because you will feel कि, okay, majority of time we talk about alpha amino acid. There could be some exception, but remember, there is no exception. Okay, there is a common name as well as there is a specific name like IUPAC name you can give, and common name is also there. Example, if you see glycine, in case of glycine, give me 1 minute. Mummy, hello. So we were talking about different type of amino acid. Example, if instead of R you have only hydrogen, then this name will be glycine. It means glycine formula will be NH2, 2 hydrogen, and COOH. This is glycine. Sania, Is it chiral compound, glycine? Tanya, is it chiral, glycine? Glycine is only optically inactive amino acid.
C is not there.
Okay. Or only optically inactive amino acid. Why is this so? Because two hydrogen are present. For being optically active, there must be chiral compound. This is not chiral compound. And how to identify a compound is chiral or not? If all the four group are different, then it is chiral. Example, COH, NH2, hydrogen. And when R value is anything other than hydrogen, then it will be chiral compound. Chiral compound is optically active. Understood, guys? This is a very popular question in competitive exam only. Glycine is the only optically inactive amino acid because it is not chiral. Okay. But sometime along with the IUPAC name, like you can name it IUPAC name, there is a common name of amino acid. Example, in case of glycine, because it is sweet in taste. Why we call it as glycine? Because it is sweet in taste. Okay, so sometime whatever the amino acid you are getting, mostly there is a common name. Example, if you see tyrosine, tyrosine is another amino acid. This tyrosine amino acid is obtained from the cheese. That's why tyro means cheese. It is obtained from cheese. That's why it got its name tyro. Tyro obtained from the cheese. And if you remember biology student in the case of um, microbes in human welfare, Anvita, Pratigya, Barinder, Chandana, Chinmay, Samarth, Sara, and Sania. Okay, only Jordan is not there. Okay. So, tyrosine means it is present in the cheese. When we name any amino acid, there is usually a symbol of three letter. You are not supposed to memorize everything. If you don't remember, it's absolutely fine. Okay, it is absolutely fine because you cannot remember all 20 amino acid. And NCERT have given this all 20 amino acid. We divide these amino acid into two parts, whether they are essential for our body or not essential. Like if body can make it inside the body or we require in the food. Based on that, you have two type of amino acid, essential and non-essential. Okay. Essential amino acid means amino acid that is required in food. Why? Because our body cannot produce. Body cannot produce it. What is non-essential amino acid, Anvita? Anvita, what is non-essential amino acid?
So, so we have to take it through food, right?
No, we have to take through food essential amino acid. No,
Oh yeah,
essential means
sorry.
body
Yeah, yeah.
can
Hmm.
synthesize these amino acid. Okay, so they are not really important in our food. So to name these essential amino acid, actually you can assume like there are 10 essential amino acid and 10 non-essential amino acid. But you must have heard some people say that there is semi-essential amino acid also. So it it is not given in NCERT, but most of teacher will teach you semi-essential amino acid. Means small amount body can produce, small amount we need from the outside, and this is arginine. But according to NCERT, it is 10 essential and 10 non-essential amino acid. Okay. Example, what are the 10 essential amino acid private team hall booked for party? Example, you have birthday party, you want to book any private uh, hall, but this is very famous in Bangalore, like team hall. So private team hall is booked for the marriage. So here how many? P V T T I M 6 HALL 10. These are the 10 amino acid essential. Don't remember all of them. Remember only 10. Remaining will be the non-essential amino acid. Example, phenylalanine. Phenylalanine. One, benzene ring. Correct. Second, valine. Third, threonine. Fourth, tryptophan. Tryptophan. I, isoleucine. M, methionine. You must have heard about methionine contains sulfur. Methionine contains sulfur. H, histidine. A, arginine. But remember, many time it is considered as semi-essential. In normal book, you will see this is the semi-essential if you read in the higher class also. And last, leucine and lysine. Leucine and lysine. NEET student, you have heard about lysine and arginine are present in the what is the histone protein, right?
Yes, sir.
Chandana, lysine and arginine are present in the histone protein, right? It is present in the histone protein. So when it is present in the histone protein, it is basic amino acid. Understood? This is important for NEET only, not for the JEE segment, lysine and arginine. But for JEE, they do ask essential amino acid. Except this, all these 10 are essential amino acid. And trust me, it is really irritating to remember these 10 amino acid and there is no logic. It's just all these are important. You should know all the name. You must have heard all the name, but you have to remember only 10. Where is the this? I'll show you this. Like just go through it. Don't memorize anything. Glycine have hydrogen. Gly or G symbol is used. Alanine, CH3. These two are very basic. Alanine, A. Valine, star means they are essential. Valine, Leucine, you are not supposed to remember anything from that. Isoleucine, arginine, semi-essential, lysine, glutamic acid, aspartic acid, glutamine, aspargine, threonine, all this. Don't remember anything. Trust me, it's not worth it at all. We don't know whether you will be asked one question or not, and you will spend a lot of time to memorize it. Got it? So don't memorize it at all. Come to the behavior of the amino acid. We know uh, protein are made up of amino acid. Barinder, what is the property of amino acid? Barinder, what is the property of amino acid? Just give me 1 minute. हेलो हाँ जी बढ़िया क्लास ले रहा था मैं 1 घंटा तो क्लास लेगी ठीक Barinder, what are the property of amino acid? Like what you know from the biomolecule of the biology? Barinder, are you there?
Yes.
हाँ. It is colorless. Why? Because simple salt. And being salt, it is a crystalline solid. Most of the solid you will study in chemistry are the crystalline solid. Right? Sarah, is it water soluble or insoluble?
So what is
Water soluble, being salt, they can dissolve in the water. And next, Sania, is it like having high melting point or low melting point?
They have a low melting point.
High melting point because of the Jupiter ion nature.
Oh, okay, sir.
Solid and crystalline solid always have high melting point. Crystalline solid always have high melting point. And when we say it behave like salt, then we are talking about Jupiter ion. Okay, we are talking about Jupiter ion, and this is extremely important for your competitive exam. Extremely important for the competitive exam. Jupiter ion is the characteristic feature of amino acid. Jupiter ion is the characteristic feature of amino acid. Samarth, what is Jupiter ion?
Sir, both cation and anion are in the same molecule.
Yes. So basically, example, we have CH3COOH. COH have acidic nature and NH2 have basic nature. So what will happen? This... Hello. Hello. हेलो नमस्ते अम्मा हाँ नंबर बताओ दे आप 90 503 84 104 एक बार दोबारा चेक करवाइए 90 503 84 104 हाँ ठीक है
ये नंबर फिट करा खाना होना को नहीं यो मिलला तो देना पर्ची पे लिख दे पर्ची पे लिख के दे दे मैंने
ठीक है मैं लिख के दे
हाँ लिख दे मैं बताओ ठीक है बैठ गया अब तो कौन मानता है
So guys
ये पेन कागज पे लिख दे नंबर मांगा था ये देखे एक कागज पे लिख दे एक कागज पे लिख दे और कर दे देंगे उसी कागज पे
just give me 1 minute
अच्छा ये कहूंगी सुन भी नहीं पे यार तो देखते होंगे सही से रह दिए सही से और अरे अरे बाबू जी इस पर लिख दे नाम लिखे रे ना है सारी नाली में जमती है सारी हो पानी तो नहीं लेगा वो पानी तो नहीं
तो थोड़ा सा स्क्रॉल अप कर देंगे वो नोट्स
ना
थोड़ा
सारी करना तो पड़ा है जमना तेरे का वो लिख दे इसका पे लिख दे इधर कर ले तेरा काम तो ये लिख दे इसका तेरे पे है इनका मिलर लीलर का नंबर
देखो मैं
ओदे भेज दिए नंबर है नहीं भैया कागजी दूंगी वो भेज दिए नहीं इसका लिख दे
ठीक है, लिख दिया मैंने। गाइस, एम आई ऑडिबल?
हैं वो आप लोग
Yes, sir.
Yeah. So what happened? COH is a acidic group and NH2 is basic group. So what will happen, Anvita, in this case? Acid will release as positive ion. What will happen to the base? Anvita,
Sir, could you repeat?
acid will release as positive ion. That's why it is CO negative. What will happen to the base? It will accept as positive ion and become the positive ion.
Yes, sir. Okay.
This positive and negative ion is known as Zwitter ion. Ion that have both positive as well as negative charge is known as the Zwitter ion. Remember, in this case, if you have aqueous solution, only then it will be shown. Because if you have solid form, then it's not possible. So this is possible only in the aqueous solution. Why? Why? Because in aqueous solution, you have medium for transfer of the H positive ion. Correct? So this is also known as the dipolar ion. A ion that have both the pole, positive as well as negative. This is known as the dipolar ion. Why? Both positive as well as negative pole. Is it positive, negative, or neutral, Pratigya? Net. Pratigya, is it positive, negative, or neutral?
Neutral.
Neutral. Because we have both positive and negative charge, net is neutral. Correct. Next, Samarth, will it show acidic behavior or basic behavior?
Sir, I think it's neutral. Like if more carboxylic group are there, it shows acidic.
It will show both behavior, acid as well as base. That's why Jupiter ion will be amphoteric in nature. So, what is the meaning of amphoteric behavior? It means it can react with both acid as well as base. And when it reacts with both acid and base, it means it can act as acid as well as base. Got it?
Yes, sir.
So, I also mentioned this already. Where is this? Glycine is the only optically inactive amino acid. Other than that, all amino acid will be optically active. All amino acid will be optically active except what, Chandana?
Glycine.
Glycine. Glycine is really important exception. Extremely important exception is glycine. So Next, Barinder, tell me which form is present in case of amino acid. Is it D form or L form? Which form is present, D or L? Example, in case of carbohydrate, there was D form, like in glucose, right? In fructose, there was L form, correct? Barinder,
Sir, alpha?
like glucose had D form, right? Fructose had L form, right?
Yes, sir.
No, both had D form. Glucose as well as fructose both had D form only, not L form. See, fructose. What is the name of the fructose? D fructose. Correct? What is this? D fructose. If you see glucose, glucose is also what? D.
D fructose.
So glucose and fructose both are D. So in this way,
L.
you have D and L form.
Liver functional dextrorotatory.
Yeah. No, not dextro and levorotatory. That is different thing. That is plus and minus. Dextro and levo. Most of people believe that this is dextro and levo. It is not dextro levo. It is configuration. So both D as well as L form can exist, and this can be asked only in your assertion region. Right, wrong statement wise, nothing else. They will give only L configuration exists in amino acid. No, both D and L can be present, but mostly naturally occurring amino acid have L configuration. Most naturally occurring amino acid are having L configuration. Remember, when we talk about D and L, we talk about configuration. Okay, L configuration mostly, not all. Mostly. Samarth, no, Sarah, when it come to the amino acid, do we have mostly alpha amino acid or only alpha amino acid?
Sir, alpha amino acids or
Only alpha amino acid. Mostly alpha amino acid or only alpha amino acid?
alpha amino. Is it alpha amino?
Only alpha amino acid. That's what we wrote here. Key in case of protein, we have only
Yes.
alpha amino acid. But when it comes to the D and L form, mostly L form, not only.
Okay, sir.
This kind of
Okay.
assertion reason are asked, and it become difficult to answer when we have assertion reason type of question. Remember, that's why people believe ki this numerical are tough. No, numerical are not tough. Numerical you can still solve. This theory question are difficult because if you know it, you know it. If you don't know it, you don't know. As simple as that. Got it? And what is the meaning of L configuration here? Samarth, I asked you twice. Tanya.
Yes. So that I'm not sure. Like here, there is no alcohol to compare. So
Yeah, there is no alcohol to compare. So L configuration is based on NH2 group will be present on left hand side. If NH2 on the left hand side, it means this is L configuration. Understood? Hmm.
yes,
Tanya, which kind of bond is present between two amino acid? Tanya, are you there? I think she's not. Am I missing anyone? Sinmaya having he can't speak.
Hello, sir.
Yes, Sania. Which bond is present between amino acid?
So peptide bond.
Peptide bond. So peptide formed is formed by the dehydration. It means removal of water, OH from COH group and hydrogen from the ammonia group. And here, which functional group will form, Chinmay? Though you have to write notes during class, but still, I will keep on asking question, Chinmay. Which functional group present between two amino acid? Bond name is peptide bond, but the functional group formed because of the formation of peptide bond is Chinmay. Are you there? My question is, which functional group is present between the two amino acid? Answer is the amide, CONH, and bond name is functional group is amide and bond name is peptide bond. And peptide bond is formed by dehydration. Formed by dehydration. Understood? Formed by dehydration. Not only this, even if you take carbohydrate, formed by dehydration, nucleic acid, formed by dehydration. This is the common thing. It is formed by the dehydration. Okay. So, like many things can form. Example, if you take glycine, here hydrogen, here CO, and NH2 of another CH3, hydrogen, and COH. You will call it as dipeptide. What does it mean? Peptide formed between two amino acid, dipeptide. An example, if you have two amino acid, what will be the number of peptide bond? 1. 100 amino acid, what will be the number of peptide bond? 99. 1 less. Okay, 100 amino acid, how many peptide bond? 99. Correct? So you can have dipeptide, tripeptide, pentapeptide, polypeptide. Polypeptide means sometimes more than 100, more than 10,000 mass, and such molecule are known as protein. So what happened? Smallest unit is amino acid. Um, amino acid. Amino acid form peptide. Peptide form polypeptide, and polypeptide form protein. So what is the smallest unit? Amino acid. What is the biggest unit? Protein. How many amino acids generally present in the protein? 100 amino acids, more than 100. Usually, protein contain more than 100 amino acid. Okay? And usually mass is more than 10,000 unit. Protein contain more than 100 amino acid and 10,000 molecular mass. So that is the definition of protein. Who will tell me? I am done with two round. Next, amides. In Anvita, does protein always contain minimum 100 amino acid?
No, I'm guessing.
No. Reason? Because you will see one exception of insulin. One exception of insulin. Insulin is a protein. This protein does not have more than 100 amino acids. This is only exception. It is a protein, and this insulin contain only 51 amino acid. Still, it is a protein. Why? Because it have well-defined conformation of a protein. Because it have well-defined conformation of protein. So question can be asked like, does all protein have more than 100 amino acid? Answer is no. There are few exceptions like insulin that have less than 100, but still it is a protein. Why? Because it have all the feature of the insulin conformation, like we will study about the primary structure, secondary structure, tertiary structure, quaternary. Everything is present in this. That's why we will say insulin is a proper protein. Got it? So we are done with the basic part. Correct? We are done the basic part. Example, how we did carbohydrate. Uh, Pratigya, carbohydrate can be divided into three major categories. What are those categories?
Monosaccharide, polysaccharide, oligosaccharide.
Huh. Monosaccharide, oligosaccharide, and polysaccharide. Barinder, what are the example of monosaccharide? Monosaccharide.
Glucose,
Glucose, fructose, ribose, deoxyribose, and many more. Do you remember how we studied glucose and fructose in detail to tell about the oligosaccharide or polysaccharide? Same way. Now we are done with the basic part of the protein, that is amino acid, their property. And based on that, we can study about the bigger structure, like how we had polysaccharide, here we have protein. Chandana, what are the example of polysaccharide? And remember, all of you have to write answer what I am asking during the class. Example, if I am asking Chandana, what are the example of the polysaccharide, you have to write in the notebook. Okay, this is my first answer, second answer, third answer. Only then you will be active during the class. And I will check everyone's notebook. Chandana,
Sir,
example of polysaccharide.
sir, I'm not sure.
First is starch. Starch is present in two forms. One is amylose, second is amylopectin. Amylose and amylopectin. Second, cellulose made up of beta glucose. Cellulose made up of beta glucose. Third, glycogen look like amylopectin but highly branched. Correct? Uh, Sania, can you tell me any other polysaccharide than uh, starch, cellulose, and glycogen other than these three?
Sir, inulin,
Inulin. Inulin is a polymer of
fructose.
fructose. This is very, very popular. Inulin, fructose. Am I clearly audible or should I use earphone?
you're audible, sir.
Yes, sir.
Clearly. Okay. So, next, Samarth, what are the example of disaccharide?
Sir, glucose.
Eh?
Sir, not glucose. Sir, sucrose, sir. Sucrose made of
Sucrose.
glucose and fructose.
sucrose made up of glucose and fructose and reducing or
Maltose.
non-reducing? Tell me everything, full
Sir,
detail.
it's non-reducing, sir, because
No.
yeah,
And bond between which one? Which carbon?
C1 and C4, sir, of.
C1, C2,
Oh yeah, C1, C2.
C1, C2
C1
is
of glucose and C2 of fructose, beta fructose.
uh,
And sir,
mal-
maltose.
maltose.
Yes, sir.
Because in yesterday we have seen question from this, right?
Yeah. Two glucoses attached.
Hmm.
Sir, and then lactose, sir. One is beta D galactose and one is beta glucose.
Beta D galactose and beta glucose and bond.
Sir, I think here C1, C4. Galactose is
C1.
C1 and C4 of glucose.
Yes, reducing or non-reducing?
Sir, this is reducing, sir.
Only sucrose is non-reducing. All
Only
other are
sucrose.
reducing.
Okay, sir.
Correct. So, so this is what like how we will revise the other topic also. So
Okay,
keep
sir.
on writing every time.
Yes.
We are done with this now. Protein class, sorry, amino acid classification is done. Essential, non-essential, acidic, basic, and neutral. Like this is not written in your NCERT, but I will give you this classification also.
So,
Ama-
could you go back to the Zwitter iron thing 1 second? For I need to see something.
where is the deuterium? Here.
Yeah, yeah, yeah.
Hmm.
Yeah, okay.
Hmm. So, amino acid, if we say there are three types depend upon the number of NH2 or COH. If it NH2 and COH is exactly same, their number is exactly same, then it will be neutral amino acid. Example, in case of glycine, there is one COH and one NH2. Then, if NH2 number is more, then you know it will be basic in nature. Lysine and arginine, basic. And if COH is more compared to NH2, then it will be acidic amino acid. It's easy to remember. Whenever you see in the end acid, glutamic acid, aspartic acid, whenever you see acid in the end, so it is acidic in nature. Okay. Then, if we classify the protein, this is not given in the biology part. Protein can be two types based on their molecular shape. Remember, whenever you classify anything, there should be criteria. Example, in this case, criteria is number of NH2 or COH. In case of essential and non-essential, uh, Tara, what is essential and non-essential amino acid?
Sir, essential amino acids, body can't synthesize it, and non-essential body can synthesize.
Yes. Okay. So their classification was based on the whether body can produce it or not. Here, number of NH2 and COH. Same way, protein will classify based on the molecular shape. How is the molecular shape? So it is two type. One is fibrous protein, and second is the globular protein. Fibrous protein and globular protein. So what is the criteria based on molecular shape? based on molecular shape. Give me 1 minute. हेलो, क्या ज्ञान? बात तो यार उस टाइम तो फिर मेरे भूल गया, मैं लग गई। मैं क्लास लेने लग रहा था, मैं क्लास के बाद करवाते रहता था थोड़ी यार। हाँ। Okay, so we have these two type of protein, fibrous and globular, based on the molecular shape. What is the shape of the protein? Correct. When it comes to the fibrous protein, chain, polypeptide chain will run parallel to each other and they will have very strong bond. When it comes to the globular protein, chain are coiled around in the spherical shape. So that is the one difference. In board, they can ask directly what are the difference. In this case, polypeptide chain, it will be run parallel to each other. Run parallel to each other. If you see in the globular protein, polypeptide chain will coil around. It's like here you have this kind of parallel to each other like this. Correct? So they will be tightly packed. Correct? They will be tightly packed. But when it comes to the globular, you will have polypeptide chain, but these polypeptide chain will coil around each other and they will form spherical shape like woolen ball, spherical shape. So which one will be stronger? Uh, Chinmay, if you compare this fibrous and globular protein, what do you think which one will be stronger out of fibrous and globular protein? Can I write in this? Hello everyone. Yes. Chinmay, am I audible? Okay. Yeah. So fibrous protein will be stronger because they are parallel to each other. So they are compactly packed. When they run parallel to each other, what will happen in this case? Compactly packed. And if it is compactly packed, what will happen? They will be stronger, harder. Correct? Okay. If you see uh, globular protein and fibrous protein, globular protein are water soluble usually because they are loosely packed, so they can form bond with the water. But if you see the fibrous protein, they are generally water insoluble. And remember these words. Generally, usually, all these words are extremely important in your competitive exam in right and wrong statement. But here we are doing the difference. Okay. Next, if you see what are the example present, then in this fibrous protein, you have in the hard structure. What are the hard structure? Example, keratin. Keratin is present in our hair, wool, silk. So fiber-like structure. Correct. You have myosin protein in the muscle. Correct. Keratin in the nails also. Globular protein are present in your enzyme usually. Usually present in most of your enzyme contain this. Also, it is present in the insulin is hormone, albumin to balance the acid base read. So, if simple language anybody compare what is the difference between these two, you will see fibrous protein it's look like parallel, compactly packed. If compactly packed, what will happen? They will have high melting point, boiling point. You can compare many things just based on this. And here, if it is like randomly packed, so it is loosely bonded. In that case, what will happen? Water soluble, low melting point, low boiling point, very soft, high activity. In fibrous protein, low activity. Understood? Any doubt? Any doubt, guys? So,
Excuse me, sir.
yes, Sarah.
Sir, I'm in hostel right now. I'll be back in 2 minutes, sir. I have to go for attendance.
Yeah, sure, sure, sure.
Okay, sir. Thank you.
Yeah. So, guys, this is how you have to compare. Key example, if this is not given in the NCERT, which one have high melting point, boiling point? Here, high melting point and boiling point. It means it will be low here. Guys, these are hard comparatively. These are soft, and many other things can be related based on this. Board will ask simple question, but competitive exam is always asked with the logical perspective. Can you correlate with the other thing which is not directly given in the NCERT? What people think when teachers are saying you have to read only NCERT, it means word to word. No, not word to word. Like if one statement is given in NCERT, you should be able to logically correlate with it. ಅರ್ಥ ಆಯ್ತಾ?
ಆಯ್ತು.
ಆಯ್ತು sir.
ಸರಿ. Okay. So this is important for your board also. Like can be asked for 3 marks. What are the difference between the fibrous and globular protein? Are you getting background noise?
Yes, sir.
Little bit or too much?
Sir, it's manageable. कोई बात नहीं.
I will use earphone because earphone are sound this have noise cancellation. That's why. Give me 1 minute. Come out.
Sir, excuse me, sir.
Yes. Yeah, Tara.
Yes, sir, sir, can you explain the last 2 points?
Yeah, sure. Am I audible, guys?
Yes, sir.
Is
Yes.
voice quality better than before?
Yeah.
Yes, sir.
No background noise.
No, sir.
Okay. So, Tara, what I said in last 2 points, because fibrous protein are parallel to each other, it means these are tightly packed. I said compactly packed. If it is compactly packed, it means they are very hard. If they are hard, they have high melting point boiling. It's difficult to boil them or melt. But globular protein are present in the spherical form, coil around each other. So they are very soft. Due to the soft nature, it's easy to boil them or melt them. That's why melting point, boiling point will be low in case of globular protein. Got it?
Yes, sir.
And because of this tightly compact nature, they are hard fibrous and globular are soft. Then Anything we missed? No. Now, what we will study further about the protein is ditto given in your biology also. That's why for JE student, they feel it very complicated. Is this true, Chinmay and Samarth?
So the structure part, sir, yeah.
Yeah. So when it comes to the structure and shape of the protein, we can study every structure of protein at 4 levels. So now we are talking about the protein structure, how we can study it. There are 4 levels: primary, secondary, tertiary, and quaternary structure of protein. First is primary structure of protein. What is the simplest structure? And it's easy to understand. Why? Because if amino acid are present in a sequence, example, first amino acid, second, third, fourth, fifth, sixth, and keep going on. So what will happen? A long straight chain of many amino acid is actually it is a polypeptide chain. So this polypeptide chain will tell you the primary structure. So Samat, what do you think? What you get? Which idea do you get through the primary structure? Do you
So
get like
some
how amino acid are
carbon chain.
aligned? Yeah, chain.
Yeah.
So primary structure give you idea about the sequence of amino acid. How amino acid are sequenced in this. Correct? And even if you heat or boil the protein, you can change secondary or tertiary structure, but primary structure will remain same. And if you change primary structure, then protein itself will change. Change in primary structure, it means overall protein will change. If you change in secondary or tertiary structure, maybe it's like, though I give the same example every time, same thing I gave in the biomolecular biology also. It's like smallest unit of the room is brick. Brick is amino acid. Sequence of brick will form a wall, secondary structure. Then, then many wall combined together will form the tertiary structure, a room. And many room together will make a complete house, that is quaternary structure. Correct? So one row of bricks will give you the primary structure. If you destroy or change the primary structure, it means automatically whole room or house will change. Nature will change. Correct? So this is like one row of brick. Multiple row of brick will form wall. Multiple wall will form uh, what what is this room and then house. So I will compare with this. Primary structure is like one row of brick. Secondary structure, multiple row of brick will form wall. Tertiary structure, multiple wall will form a room, one room. And quaternary structure, multiple room will form the complete house. Make sense? Okay. Primary structure is done. Then you will have secondary structure. You get more question from the secondary structure compared to the primary because primary is very easy to understand. Secondary structure of the protein.
So when we talk about this secondary structure, same single chain, straight chain will increase its length and it will coil like this. Uh, So in this, you will get to know about the shape of the protein. Okay, shape of protein or polypeptide chain. Because example, in case of primary structure, it is straight always. So primary chain tell you about the sequence of amino acid, like what will be the first amino acid, second, third, fourth, name of the amino acid. In secondary, when same primary chain become very big and change its shape, the shape is explained by the secondary structure. Correct? This secondary structure is present. This secondary structure is present in two forms: alpha helical form, alpha helix, and beta pleated form, alpha helix and beta pleated form. These are the two forms of secondary structure only. Sania, what is the difference between these two, alpha helix and beta pleated structure?
Sir, in alpha helix, it has a coiled structure,
Hmm.
whereas in beta pleated, they are arranged in parallel sheets.
In beta plated, it's like you have coiled form, then you extend it, you stretch it out. It will zigzag. Like alpha helix is like same, this, correct? Beta plated is like same thing, but then you stretch it out and it look like this. Beta plated sheet, like draper, like your curtain. In your window, you have curtain, this curtain shape, not properly helical, but still it is look stretched form of the helix. Okay. So if anybody asks what is the common structure present in the secondary structure, answer is the alpha helix is very common. Is this Jordan? No, Chinmay. Chinmay, am I audible? Any network issue? Yeah. Okay, good to go. So if anybody asks you which one is common out of these two, alpha helix is very common compared to the beta pleated helical structure as like spiral winding. Correct? In this alpha helical, you will see there is right hand screw helix only, right hand fashion. In biology, you know this. This is the only structure how it is coiled. Example, if you take this DNA double helix model, correct? Whether you take amino acid sequence in the secondary structure, it is always right hand fashion. It is always a right hand fashion, correct? It is always right hand fashion. And you will have lot of here you will have lot of hydrogen bond as well. Hydrogen bonding is very common because of the coiling. Hydrogen of one can interact with the oxygen of other because you have COH, correct? You have COH. What happened in case of beta plated structure? This beta plated look like sheet. It look like sheet. Okay. And in this structure, it look like you stretch it out side by side. Stretch out side by side. And this maximum extension, it look like the draper. Same thing. You have alpha helix, then you stretch out. Any doubt, guys, in this?
No, sir.
like fold of drapery. And fold of drapery is simply fold of your curtain. Fold of your curtain, how your curtain fold look like. And it is beta plated sheet. Secondary structure done. Tertiary structure, further coiling. Tertiary structure, it means further coiling of the protein. So what happened? Example, in case of primary structure, you have straight chain. Secondary structure, you have folding. In tertiary structure, you have folding further. This is tertiary structure. Here, you talk about the overall folding of the chain. Polypeptide chain, correct? Chain. What is the benefit of this? Here you can see proper shape. It could be fibrous protein or it could be globular protein. Both are possible. So here you will get proper fibrous protein shape or globular protein shape. As of now, it was simple chain. Now it is more folded. So you can check the shape. Correct? If anybody asks what are the bond present in the tertiary structure, hydrogen bond is very common. Disulfide bond you have seen in case of fibrous protein, and van der Waals force because of the mass always exists. Electrostatic force if you have positive and negative ion like ammonium ion or acetate ion. So these are the different kind of force you will see in case of tertiary structure. Force, it means we are talking about bond. And what are the bond present in this case? Hydrogen bond. What else? Disulfide bond. Van der Waals force because of the what should I say? Because of the mass, it is always there. Van der Waals force, or it could be electrostatic force also because of the ion, positive and negative ion. Any doubt, guys? In biology, we talk about this tertiary structure is actually biologically active, not given in the chemistry. It is biologically active. It means if any protein want to work in our body, this protein minimum should have tertiary structure. It means secondary structure cannot work. You need minimum tertiary structure, biologically active. Example, protein acting as an enzyme. So for being enzyme, you need minimum tertiary structure. Correct? You need minimum tertiary structure. This is primary, this one is secondary, this one is tertiary. Because biologically active means it have active site for binding with the any substrate because it can act as an enzyme. It can act as an enzyme. Quaternary structure like further folding. Here you have one chain, there you can have multiple chain, and this is quaternary structure. Quaternary structure, and I am extremely bad in the spelling mistake. Okay? Even it could be wrong. What is the wrong in this? I think R is not there, right? Quaternary structure. What does it mean? In primary, you have straight chain. Secondary, coiling. Secondary can be present in the two form alpha helix, beta plate. Tertiary, further folding. Tertiary structure have hydrogen bond, disulfide bond. And quaternary, multiple chain, like tertiary structure. 1, 2, 3, 4, like this. Okay, this is quaternary structure. When many polypeptide chain are present. Example, up to tertiary, you are talking about only one chain, but very long. too much folding. But in quaternary, you have multiple polypeptide chain. Example, in this quaternary structure, you have 4 polypeptide chain. 1, 2, 3, and 4. Okay. Each chain is known as subunit. Quaternary structure have 2 or more polypeptide chain. Each polypeptide chain is known as subunit. Each chain will be known as subunit. And 3D arrangement of this polypeptide chain give you quaternary structure. So this quaternary structure will have special arrangement, means 3D arrangement. Special arrangement means 3D arrangement with each other. How they are present along with each other, it is the quaternary structure. Example, hemoglobin. If you, here it is not. So if you take hemoglobin, HB, you will see hemoglobin have 4 chain. What are those 4 chains, Chandana?
Sir,
What are the 4 chain? What are the 4 chain present in hemoglobin?
sir, H, alpha,
How many alpha, how many beta?
I'm not sure.
Pratigya?
Alpha to beta.
Yes. 2 alpha chain and 2 beta chain. Total 4. So hemoglobin have quaternary structure of protein where 4 chain are present with respect to each other and form quaternary structure. Okay, remember 4 chain. Okay, and what else we left with? Enzyme, vitamins, nucleic acid. Now we can finish in one class easily with question. I told you in 2 class we'll finish everything. So I will do denaturation of the protein. Topic will be done, and then we can solve some question. Any doubt, guys, as of now? Because we are done with the majority of the protein. Okay. So if anybody asks what we did in the protein, we did protein. Simple meaning is primary importance. They are highly important. That's why they have primary importance. That is the literal meaning of the protein. Protein made up of amino acid. Amino acid have NH2 group, COH group, hydrogen, and one R group. Based on this R group, we have 20 type of amino acid. This amino acid have deuter ion also, positive and negative ion both. Amino acid can be different type. It could be essential and non-essential. It could be acidic, basic, and neutral. Correct? And these amino acid will form protein by the peptide bond. Protein can be 2 type based on the type, like whether they are water soluble or insoluble, based on their shape. Fibrous protein and globular protein. Fibrous are parallel to each other and they form spherical shape like this. Globular. Protein have 4 shape. Primary will give you sequence like straight chain. Secondary give you shape. Shape like this folding alpha or beta. alpha helix, or beta pleated. Third, tertiary structure is responsible for all the activity, biological activity. Like if it is active in the human being, it is minimum tertiary structure. Further folding like this. And quaternary structure, when you have multiple subunit, two or more subunit. Example, in hemoglobin, you have four, two alpha and two beta. Tell me, did we revise whatever we did today in just 2 minutes?
Yes, sir.
So, though I told more than 1,000 times, I guess, but still I would say, whenever you take a lecture of 1 hour, 1.5 hour, 2 hours, always try to summarize it in 5 or 10 minutes. If you can do it, it means you understood everything. If you can't do this, means you did not, like, you are not well versed with the topic. Last topic of this protein is denaturation of protein. Sania, what is denaturation of protein?
Sir, the destruction of the uh, structure of protein due to high temperature or pH change.
Ah, so if you have tertiary or quaternary structure, and then you change the pH or change the temperature, and real nature of the protein will be destroyed. D means destruction. Naturation means nature. Destruction of the nature of the protein. Denaturation. Destruction of the nature of the protein. It could be because of the chemical change or physical change like temperature. Okay, this is the denaturation of protein. Protein lose its biological activity. Its biological activity. Samarth, can you give me some simplest example where we, protein lose uh, its biological activity in real life? When you boil the egg, you
Yeah.
destroy the nature. That's why it was soft in the early stage, but after boiling, it became hard. Correct? When bacteria act, so what will happen? What is the reason? Due to physical change, example temperature, and because of the chemical change like pH. Samarth, any other example in real life where you felt ki, yes, there is a denaturation of protein?
Sir,
Samarth,
burning something.
example, when we have fever, we don't want to eat anything because our enzyme act in specific condition. Like they have optimum temperature 37 degrees Celsius. When we have fever, temperature might become 38, 39, or 40. So our enzyme will denaturate. When enzymes are not available for the digestion, there will be no digestion. That's why in fever, we don't want to eat. Make sense? Very basic example of the denaturation, right? Understood?
Yes, sir.
Example of this denaturation. Example, if you boil the egg, boiling of egg will change its shape. Or curdling of milk, when milk convert into the curd. Correct? In both the situation, what will happen? There is a change in the chemical nature. How? Because milk was liquid and curd become solid like. What is the reason? Lactic acid is responsible for this from bacteria. Bacteria produce lactic acid, and this lactic acid convert milk into curd. Correct. What happens when you boil the egg? This egg white will coagulate, and that's why this egg white coagulate becomes solid, and we can eat it properly. Correct. What is the reason? We understood that uh, because of the change in the physical or chemical condition, there is a denaturation of the protein. What is the reason? Chemical reason. Answer is whenever you increase the temperature, then globules, globular protein, mostly globule will unfold. When globule unfold, what will happen? Helix will uncoil. And when helix is uncoiled, then protein will lose activity. Its activity. Understood? And when we destroy the protein activity, remember, you can damage secondary or tertiary structure, not primary. During denaturation, if you destroy the nature, denaturation, during denaturation of protein, you will see secondary or tertiary structure will be destroyed because they are very sensitive to the change in temperature, but primary structure will be intact. Primary structure will be intact in this case. Got it? Primary structure will be intact in case of denaturation. Last thing in the denaturation is what is native protein? Who will tell me? Chandana, what is native protein? Any protein present in biological system, it means living being, and have one like 3D structure, like tertiary or quaternary structure. And in this structure, it has some biological activity. Due to this tertiary structure, 3D arrangement, what will you see? There is some biological activity of the protein. Such protein is known as native protein, native of our body. A protein that is present only in our body, have some function, is native protein. Understood? Topic is done. Now you can solve 90% question of this chapter. Though I have limited time, tomorrow we'll solve more number of questions because majority of part is done. Now we have only more theory part. We can easily finish in half an hour and 1 hour only for question. But still, we can solve some question.
So
Mind
we're
by
doing
mind.
the remaining thing today itself, like right now.
Majority of things are done. Only vitamins is one important thing. Nucleic acid and enzyme is not so important.
So tomorrow we'll do.
Yes, tomorrow we'll finish. It's almost done. Like 90% portion exam wise is done.
Okay.
Example C, guys. Sometimes they directly ask question, what I can do? This is NEET JEE 2025. They are asking direct, what is the essential amino acid? In this case of essential amino acid, where is this? See this question. Alpha helix and beta pleated. Seed structure of protein are associated with. Samarth, what would you say? Very difficult, na?
Very direct, sir.
See, guys, this is what they are
Tertiary
asking.
structure. Yes.
Secondary structure, alpha and beta plated, right?
Yeah.
I can show you more question, and all these are 25. Starch give galactose. Sarah, is this correct?
No, sir.
No. Starch give glucose and fructose. Uh, Sania, cane sugar give equal amount of glucose and fructose. Is this correct?
Yes, sir.
Yes. Cane sugar means sucrose. Sucrose give equal amount of this. Barinder, milk sugar give glucose and galactose. Is this correct?
Yes, sir.
Yes. Milk sugar means lactose we are talking about. Lactose. Uh, Pratigya, amylopectin give glucose and fructose. Pratigya, no, because amylopectin contain only glucose reserve or food. Anvita, amylose give only glucose.
True.
True. Amylose, amylopectin, glycogen, cellulose all have only glucose. Whatever polysaccharide we have in chemistry syllabus, it is only glucose. So there's no doubt. Then we did this already. Did this already. I have seen one more question. See. So first write PVT Tim Hall. PVT Tim Hall. Do we have V in this?
Yes, sir.
Yes. Second, what is V stand for? Valine, correct? What V stand for? Valine, so it is there. What is this P stand for? Phenylalanine. Phenylalanine. So this is not possible. Lysine, lysine, leucine. Both are there. Lysine and leucine. This is there. This is there. Barinder, threonine. Barinder, threonine is there.
Yes, sir.
Yes. Who else is left? Samarth, tyrosine is there. No.
Sir, tryptophan is
Why?
there for another T. Tyrosine is not there. Tyrosine is
Yes,
that
that's why I
cheese
wrote this.
protein.
You have only two T. One is threonine and tryptophan. So that's why he. That's the reason why I made it for essential amino acid, because for non-essential, it like whatever is not essential, it is non-essential. N- So according to NCERT, essential 10, non-essential 10. But in higher classes, essential 9 and 1 semi-essential arginine. Got it? See what kind of question? Ditto NCERT question, nothing else. See, Anvita, all naturally occurring amino acid except glycine containing one chiral center.
So wrong.
they might have more than one chiral center also. One is correct, but they can have more than that. So that's why it is wrong. Second, Varinder, amino acid cysteine can easily dimerize due to the presence of SH group. Remember methionine and cysteine. I will write in your notes. Methionine and cysteine are two amino acids that have sulfur in it. I wrote methionine, but I think I forgot cysteine. So it contains sulfur. S sulfur. Okay. So, whom I asked, Varinder, amino acid cysteine can dimerize due to presence of free acids. Sulfur is present in the cysteine. Yes. Okay. Even sometime if you are not sure about one, you can eliminate. Glutamic acid is the only amino acid that contains COS group at the side of the chain. No, almost have, not almost, all amino acid have COS group. Okay, but when it comes to the side chain, even aspartic acid will have this, right? Glutamic acid, aspartic acid, anything that end with the acid will have more number of COH group. In side group, there will be more COH. Correct? That's why this is wrong. And Sarah, all naturally occurring amino acid are optically active.
False, sir.
Um, this is not, glycine is not correct.
Yes.
Yeah. So done. So you can see, you can solve every question from NCERT or by elimination method from NCERT. And that's why whatever you will say, except one or two questions, you can solve any question from this chapter now because majority of question asked either from the protein or carbohydrate. There are few questions from the this also, but that is very less in comparison. We did all this stuff. And remember, all these are the JEE Main 2025. That's why I believe you should write this ribose sugar is pentose sugar, correct? Does it have pyranose form? Samarth? No.
No,
Glucose have
no, sir.
pyranose. Glucose have pyranose form. Deconfiguration, yes. D-ribose. Reducing sugar, yes.
S- uh, Sir, only glucose has pyranose form, right?
Yes, glucose have pyranose form. Glucose or glucose-like structure, 6 carbon. But I think this statement is not complete. We don't know what these are. Reducing sugar when free. Like if you, because if you take ribose is like this. Here OH, here OH, OH, and this. So there are 5 carbons, CH2OH in the ribose pentose sugar. So when free, it is not complete, I think. People who made the paper made mistake. Not JEE, but people who made this paper, this PDF. E. Alpha anomeric form is present. Yes, alpha anomeric form is present. What does it mean? Carbon can have OH on the right hand side alpha, or OH on the left hand side beta. Yes, guys. Done. So tomorrow we'll solve a lot of questions because half an hour 45 maximum a minute will take for to complete this chapter and then question. Tomorrow everything will done from the biomolecule, including NEET question, JEE question, and other stuff. So read this properly.